My presentation here is Innovative Antibiotics and Their Clinical Niches. My name right below, and just a quick thank you to Courtney as my content expert, who was a huge help over the last couple of weeks. So just a quick overview of this presentation. For the beginning, I'll just give a quick introduction of some of the concepts I'll be discussing and a couple of the common mechanisms of antibiotic resistance that we tend to see clinically. Part 3 will focus on 4 different novel antibiotics that I've chose to focus on based on supposed relevancy to practice here at UW. And finally, some key takeaways for clinicians who might be watching. As a result of this program, a couple of learning objectives. Participants should be able to identify pathogens of critical priority and their most common resistance mechanisms. They should be able to match novel antibiotics to those resistance mechanisms and to summarize landmark clinical trials that led to their approvals through the FDA. So what is antibiotic resistance? Essentially, random mutations in targeted pathogens can help those pathogens continue to survive. And as a result, those pathogens will pass on the gene that made them resistant in the first place. Multi-drug resistant pathogens are defined as resistance to at least 3 different classes of antibiotics. These are most common in the hospital setting and are a major contributor to infant mortality and deaths through infectious disease across the country. Over the last 3 decades, unfortunately, there's been a decline in novel agents and a corresponding sharp increase in resistance as we enter what's known as a post-antibiotic era. And I'll talk a little bit about why that is. So as we enter this post-antibiotic era, the World Health Organization in 2017 released a list of what they call their leading antibiotic-resistant bacteria in order to help aid in surveillance and data collection on these organisms, help control infections across the country and across the world, and to continue research into what we can do to prevent those infections from spreading further. And they prioritize these pathogens as critical, high, or medium priority in terms of risk. Critical priority pathogens are obviously the most important and the most closely watched in modern society because related infections are most likely to be lethal as compared to the high and moderate priority pathogens. These pathogens include Acinetobacter species as well as Pseudomonas aeruginosa. C. diff is a significant causative factor, and Enterobacteriaceae family as a whole. And I've listed those down below. We'll go into the important ones individually. However, it's also important to note that carbapenem-resistant Enterobacteriaceae infections are currently on the rise, and these are a significant target of new antibiotics that are being released and developed. High priority pathogens, so this is a step down. We have vancomycin-resistant Enterococcus, MRSA or methicillin-resistant Staph aureus, Helicobacter pylori, which is a major risk factor for stomach cancer. We have Campylobacter, which is responsible for food contamination, and Salmonella, which is also responsible for food poisoning. And then we have Neisseria gonorrhea, which is a causative factor in STIs. Since resistance is on the rise among our current antibiotics, naturally new molecules are needed to make sure that we have an existing utility to our current antibiotics and that they don't continue to develop resistance as time goes on. Unfortunately, the companies with the most resources to engineer and market these antibiotics, meaning large companies with large pools of resources and research, are not financially interested in these ideas. And the reason for that is because there's a negative investment return on these antibiotics. Hospital facilities don't tend to use them as often as they would their go-to antibiotics, simply because there's minimal data on them because they're only recently being licensed and approved for certain indications. And there's also minimal incentive to use them since they tend to be restricted to inpatient use and restricted to circumstances in which cheaper and more effective and more broadly studied antibiotics are available as an option. So the burden then falls on smaller startups who are now proposing newer approaches and a stronger commitment to antibiotic stewardship. There have been 8 innovative antibiotics approved within the last 5 years, and the majority of these were developed from molecules and structures that already exist, targeting CRE, which is carbapenem-resistant Enterobacteriaceae, which I'll be mentioning in that acronym moving forward, and other critical priority pathogens on that first list I showed you. Innovative antibiotics are antibiotics that do not show cross-resistance to existing antibiotics. And if that information isn't available, innovative antibiotics are designated by either a new scaffold or central structure known as a pharmacophore, a new target or binding site, or a new mechanism of action. And the World Health Organization outlined 5 different objectives of their action plan on resistance as below, which I have as improving awareness of resistance, strengthening the quantity and the quality of the data that we have to study them, reducing the incidence of infection, optimizing use of antimicrobials, and increasing our investment in drugs, vaccines, testing, and other diagnostics. So now just to run through a couple common mechanisms of resistance that we'll see the innovative agents that I'll discuss attempt to exploit and triumph over. So from left to right here in this diagram, we have changes in cell metabolism and binding site, antibiotic breakdown, antibiotic entry, and antibiotic efflux. Each of these known resistance patterns contributes to the need for novel agents, and I'll describe each one of them moving forward in greater detail. I did mark each one of them with a symbol, as in gram-negative and gram-positive, where they are most commonly seen in terms of bacteria populations. So in our first resistance mechanism, bacteria produces proteins that will break down the antibiotic so it can't do its job anymore. And a really great example of this, and one that I'm sure many of you in our audience are familiar with, is the hydrolysis of beta-lactam antibiotics by beta-lactamase enzymes, which the bacteria produce to withstand the activity of penicillin. Enterobacter species also have the ability to produce expanded spectrum beta-lactamase, or ESBL, which break down even more advanced types of beta-lactams. I've also provided a list of other inactivating enzymes, although they're less clinically relevant to this presentation. Other bacteria can develop random mutations in the part of the cell that an antibiotic would target, like the penicillin binding protein that beta-lactams target within MRSA. And this is so that the antibiotic cannot bind as well and inhibit the function of that protein. And this also occurs in azithromycin and other macrolides. And that's where a methylation pattern mutation in the ribosome makes it harder for macrolides to bind. So the ribosome therefore continues to carry out its function of making protein, and the macrolide does not work anymore. Some bacteria are also able to create structural changes in the cell membrane itself, often changing the porins through which antibiotics might pass to restrict their entry. And this can limit molecule size, degree of hydrophilicity, and the charge of the molecules that are allowed to pass through. And this is responsible for Pseudomonas resistance to imipenem. Drug efflux pumps may also arise as a result of mutations in cell membrane proteins and might more quickly remove antibiotics from the cell through active transport. And also, if a certain metabolic action is disrupted by an antibiotic, as this occurs in sulfamethoxazole trimethoprim or Bactrim, bacteria can find alternative pathways to produce those necessary metabolites, such as within the folic acid cycle. So here's our first learning check, and you can answer this in the chat. Which of the following is not a mechanism of resistance that has required the development of novel antibiotics in recent decades? And I have the options listed here as restriction of antibiotic access, change in binding site, production of inactivating enzymes, and upregulation of influx pumps. All right, so I have the correct answer here as D. A through C are documented mechanisms, but D is not a correct answer because efflux pumps would be the documented mechanism. So pumps that push antibiotics out of the cell rather than bringing them in. So for each novel antibiotic, of which I'll discuss 4, I'll give a brief introduction and then talk about targeted pathogens, how the antibiotic evades resistance patterns, safety profile, and I'll finish up with a quick summary of the landmark trials and takeaway points. And the first antibiotic we'll discuss is delafloxacin, branded Baxdela. So delafloxacin was first manufactured in response to rising resistance within the fluoroquinolone class and use-limiting adverse reactions, which I'll talk about in the safety section. Of note, structure-wise, this is the only non-zwitterionic antibiotic in the class. A zwitterion is an ion that has negative and positive charges on different sites within the same molecule, but an overall net charge of 0. And we'll talk about why that's relevant. This was first licensed by Melinta Therapeutics and approved by the FDA in 2017 for the indication of skin and soft tissue infection. At UW, we have a list of restricted qualities here. So Ordering of delafloxacin is restricted to an infectious disease attending or a fellow. Use is limited to 24 hours prior to discharge only. And before it can be initiated, you have to verify that the patient can access this antibiotic reliably once they are an outpatient, meaning that a test claim should be sent and approved and documented whether or not the patient can afford this medication. Because out of pocket, this medication costs $85 per 450 milligram tablet and $170.28 per intravenous vial. Targeted organisms include both Staph aureus, methicillin susceptible and resistant, Enterococcus species, Streptococcus species, and Escherichia coli, especially in surgical sites. It should be noted that although delafloxacin does technically cover Pseudomonas, it has weak coverage and therefore is not a great empiric choice if you are starting a patient on new therapy for Pseudomonas. So for a mechanism of evasion here, delafloxacin is within fluoroquinolone class. It's the only fluoroquinolone that has equal affinity for both DNA gyrase and DNA topoisomerase 4. And so in that sense, resistance to delafloxacin requires a higher number of mutations than earlier tetracyclines, which are selective for one of the two enzymes. DNA gyrase arises through genes GYRA and GYRB, and topoisomerase 4 arises through PARC and PARE. And these genes are found in both Staphylococcus aureus and gram-negative bacteria classes as a whole. And so that makes resistance to delafloxacin less likely in these species, since they again would have to have a higher number of random mutations coincide to produce both gain-of-function mutations in DNA gyrase and topoisomerase that prevents delafloxacin from acting on them. Delafloxacin is also active in acidic mediums and therefore is effective for skin infections, which is what led to its first approval, and it is an anion at neutral pH. As a quick safety profile, all black box warnings, of which there are clearly a multitude, a bit of a laundry list here, all black box warnings that apply to fluoroquinolones also apply to delafloxacin. And these include, to name a few, central nervous system effects, psychiatric reactions, tendinopathy, and superinfection. Peripheral neuropathy and C. diff-associated diarrhea, however, were equally frequent in the control group. So these are not significant risks, despite the fact that the warnings are carried over into this new agent. There is also no QT prolongation or phototoxicity seen with delafloxacin in comparison to other fluoroquinolones within the clinical trials that have been done so far. And all labeled adverse reactions are estimated at less than 2% incidence. So that includes both mild and severe. Delafloxacin is a mild inhibitor of the hepatic enzyme 3A4, so important to watch for drug interactions, although few clinically significant interactions have been documented thus far. This is what I would consider the landmark trial that led to the approval of delafloxacin, and it's entitled Efficacy and Safety of Delafloxacin Compared with Vancomycin plus Aztreonam for Acute Bacterial Skin and Skin Structure Infections. So of note here, they're comparing delafloxacin with a very broad spectrum of coverage, vancomycin covering a vast majority of gram-positive organisms, and aztreonam covering the gram-negative side. So the objective of this trial was to establish non-inferiority versus vancomycin plus aztreonam for skin and soft tissue infections and to compare the safety of the two regimens. This was a phase 3 multicenter randomized double-blind active controlled trial enrolling 660 patients, so a relatively large number, and it compared delafloxacin versus these two agents twice a day for 5 to 14 days. Endpoints are listed below. In erythema reduction, investigator-assessed cure, MRSA eradication, and adverse reactions. In erythema reduction, this was a non-inferior endpoint, as well as investigator-assessed cure were also similar at early and late follow-up between the two regimens. MRSA eradication, delafloxacin's rate was 100% eradication, with only 98.5% for the alternative regimen, the control group. And adverse reactions were similar between groups, and more adverse reactions leading to discontinuation were seen with vancomycin and aztreonam. So in conclusion, the authors were able to state that delafloxacin is non-inferior to vancomycin and aztreonam at 48 to 72 hours and is well tolerated as monotherapy for this particular infection type. My brief summary of delafloxacin here. Mechanism of action: this is a new synthetic fluorocycline because it has a fluorine group, and it equally inhibits DNA gyrase and topoisomerase, leading to its balanced activity between gram stains. Approved indications are skin and soft tissue infection. It can also be used for community-acquired pneumonia, but this has been sparsely studied and isn't a strong niche at this time. Of note, although most fluoroquinolones are bioequivalent in oral and IV forms, delafloxacin is not. It's 450 milligrams orally or 300 milligrams IV Q12 hours, and it comes in those two formulations only. 58.8% bioavailable. Cost would be $170 per vial, about $85 per tablet, and it is restricted here at UW under the criteria I underlined. The next antibiotic I want to discuss a little bit is the combination product meropenem-vaborbactam, which is branded as Vabomere. Of recent decades, gram-negative infections have been increasing, especially carbapenem-resistant Enterobacteriaceae, or CRE. This is a very invasive and very aggressive type of infection. Unclear data on mortality between 26 to 44% as of today. And resistance to these type of bacteria comes from carbapenemase, which is a type of beta-lactamase that these bacteria can produce. And unfortunately, it's unaffected by previous beta-lactamase inhibitors that we've been able to manufacture, including clavulanate, tazobactam, and sulbactam, which are all found in products that we use here at UW. There is no first-line antibiotic for carbapenem-resistant Enterobacteriaceae. And products that we know may be effective, such as polymyxins, aminoglycosides, and colistin, have significant toxic effects like nephrotoxicity and ototoxicity, and also require therapeutic drug monitoring. Many beta-lactams, because of this beta-lactamase production, are ineffective by themselves. And that's because the beta-lactams, while they act through binding this penicillin binding protein, interrupting the synthesis of the cell wall and the cell membrane, expanded spectrum beta-lactamase also confers resistance to a whole broad variety of beta-lactams, so now we need new types of beta-lactamase inhibitors to prevent this resistance mechanism from moving forward. Meropenem-vaborbactam was manufactured from Rempex Pharmaceuticals. Meropenem as an monotherapy is a broad-spectrum carbapenem, and vaborbactam is a new beta-lactamase inhibitor based on a boronic acid pharmacophore. In vitro experiments were used to find the best substitutes on an existing beta-lactamase inhibitor to enhance the activity of meropenem against CRE. This is dosed 4 grams IV, so 2 grams of each product, every 8 hours for 5 to 14 days, and it's only been approved thus far for complicated urinary tract infection. At UW, this is restricted to an ID attending or a fellow, and the patient needs to have a confirmed carbapenem-resistant pathogen or a history of one, and you can only use Vabomere if an alternative agent is not possible to use. And this would be a cost of $246.80 per 2-gram vial. And so this would be about $20,000 out of pocket for a 14-day course, which is pretty significant. So different agents that each component of Vabomere targets. Meropenem has broad coverage by itself, including various gram-negative rods, including Enterobacteriaceae, Enterococcus, Streptococcus, and staph species, as well as several anaerobes. However, resistance to meropenem has been trending up, as I mentioned. Vaborbactam, as a beta-lactamase inhibitor, has no antimicrobial activity on its own. However, it does counter various types of broad beta-lactamase, and I've listed these here. However, we might find that vaborbactam might still encounter bacteria to which, bacteria may still become resistant to vaborbactam due to expanded spectrum beta-lactamases, which continue to evolve and mutate, as well as efflux pumps. And this is most commonly found in Acinetobacter and Pseudomonas species. As for clinical efficacy of Vabomere, vaborbactam did enhance meropenem efficacy for CRE. Tango 1 was the first major trial, and this was a randomized phase 3 study in which meropenem vaborbactam was administered to patients with confirmed or suspected CRE hospital-acquired pneumonia and or bacteremia. The control groups in these trials with the Tango series are a little bit hazy because they only were able to use the quote-unquote best available therapy, meaning the agent that the clinician was able to choose, because there is no, at this point, single specified agent that works best against CRE. So the control group was comprised of all patients who received this list of agents here below, and the treatment group, which was Vabomere monotherapy, showed a significant reduction in mortality. Meropenem is stable to penicillinase and cephalosporinase due to the presence of a 6-trans hydroxyethyl group. And vaborbactam evades class A and C beta-lactamase as well as carbapenemases, which meropenem alone cannot do. And the reason it's able to do this is because boron, which is part of the structure of vaborbactam's pharmacophore, it mimics a tetrahedral intermediate that's formed when meropenem interacts with the beta-lactamase molecules within the bacteria. And that locks up the beta-lactamases so that it can no longer break down meropenem. And vaborbactam itself is able to enter the outer membrane of gram-negative rods, especially Klebsiella, via certain porins, K35 and 36. So it's of a structure that easily allows it to slide into the bacteria. Clinical studies thus far have demonstrated fairly solid tolerability of Vabomere in comparison to control groups. All adverse reactions were ranked between 3 to 9% incidence and included headache, mild infusion site reactions and rashes, phlebitis, and diarrhea. Warnings, hypersensitivity, including all, so hypersensitivity carries over through all beta-lactams. So this is a warning that applies to all beta-lactam antibiotics, as well as the central nervous system effects that are seen with carbapenems and sometimes cephalosporins. Superinfection is also possible, including C. difficile-associated diarrhea. So these are associated with broad-spectrum antibiotics. They've been attached to Vabomere as well as warnings. So Tango 2 is likely the landmark trial here. This is entitled The Effect and Safety of Meropenem Vaborbactam versus Best Available Therapy in Patients with Carbapenem-Resistant Enterobacteriaceae Infections. And they use BAT as an acronym for Best Available Therapy. So this study compared the efficacy and safety of Vabomere monotherapy versus best available therapy for CRE. This was a phase 3 multinational open label trial from 2014 to 2017. And only included 77 patients with CRE, 47 of which were actually confirmed. So of note, a slightly lower number of patients were confirmed with CRE versus suspected. And these 47 patients, or 77, apologies, were randomized with 2-to-1 to meropenem vaborbactam, favoring the experimental group. And they received 2 grams of each component over 3 hours every 8 hours for 7 to 14 days. And again, this trial compared Vabomere to best available therapy. The results showed more likely clinical cure, lower mortality at day 28, and less nephrotoxicity versus best available therapy for carbapenem-resistant enterococcus. So overall, to summarize, the mechanism of action for meropenem vaborbactam, we have a peptidoglycan or cell wall synthesis production inhibitor and a beta-lactamase inhibitor combined in one product. And these help to protect the beta-lactam of meropenem from degradation by beta-lactamases and carbapenemases. Approved for community-acquired UTI, dosed 4 grams every 8 hours, and cost, as we mentioned, could be up to $20,000 per treatment regimen and restricted at UW for those reasons. And I have question number 2 here for learning check. A 53-year-old patient is diagnosed with Klebsiella pneumoniae pyelonephritis. This is her 3rd hospitalization for a UTI in 2 months. Susceptibilities are currently pending. And if the team asks for pharmacy's recommendation, what would you do? Answer A would be to recommend meropenem vaborbactam, refer them to the infectious disease fellow so that they can order meropenem vaborbactam, check to see if the patient has a recent history of carbapenem-resistant Klebsiella, or recommend meropenem. So I had answer C listed as the correct answer. For A, we may not need to use meropenem vaborbactam in this case, since meropenem by itself might suffice. And for B, the same answer here. We may not need to use such a broad and expensive agent at this time. C would be the correct answer because if resistant isolates are resistant to meropenem, then adding vaborbactam would be appropriate and may create an effective combination. And as for D, since the patient has had several hospitalizations for similar infections, it would probably be best to check previous susceptibilities before we choose an agent. And the third agent I would love to discuss with you here today is eravacycline, or Xerava. Eravacycline is a synthetic fluorocycline, which is a tetracycline derivative that was produced by Tetraphase Pharmaceuticals. It's indicated right now for complicated intra-abdominal infections, which are characterized by extension of abdominal infections into the peritoneal cavity. And this can cause localized peritonitis that's associated with increased mortality and often needs to be treated as soon as possible to control the source of infection. Tetracyclines do come in several different generations in order of manufacturing and approval. First generation is considered tetracycline. Second generation, we have doxycycline and minocycline, which are likely the agents that we see most in this hospital setting. And third generation, a very broad agent called tigecycline. At UW, eravacycline is restricted to an ID attending or fellow via consult or pager and costs $63.60 per 50 milligram vial. Targeted organisms for eravacycline include Enterococcus species, including VRE, staph species, including MRSA, strep species, various anaerobes, and multi-drug resistant gram-negative rods, including CRE. Of note, similar to the previous antibiotic we discussed, we have seen poor activity against Pseudomonas and insufficient data at this time against Acinetobacter. Although it does cover Acinetobacter, we don't have sufficient data to recommend it as a first-line option. Eravacycline has successfully been seen to evade efflux pumps, which is the most common resistance mechanism within the tetracycline class. And this affects first-generation tetracyclines more than it affects second or third generations. There are various types of efflux pumps that can help push tetracyclines out of the bacteria before it can perform its function, including TET A and TET B in gram-negative bacteria and TET K and TET L in gram-positive bacteria. And these induction TET genes on plasmids lead to membrane protein synthesis, and those in turn reduce the concentration of antibiotic within the cell. Eravacycline also bypasses the resistance mechanism of ribosomal protection proteins, which also affect the first and second generations more than they affect the third generation. And these weaken interactions between tetracycline and their ribosomal binding site, and leading to reduced susceptibility to earlier generations. The way that eravacycline is able to evade efflux pumps is the addition of a fluorine group, which is what leads to its classification as a fluorocycline. And this also grants it its activity against gram-positive and negative pathogens that are often resistant to our first and second generation tetracycline agents. Multi-drug efflux pumps have been observed that still recognize the third generation tetracyclines, but fortunately they haven't been shown to be significant enough for clinical resistance, meaning that the antibiotics still work, even though the efflux pumps are able to push the antibiotic out partially. Tetracyclines also bind reversibly to the 30S subunit and inactivate the aminoacyl tRNA complex, while eravacycline binds at multiple attack sites, which leads to stronger binding and a more stable complex that ribosomal protection proteins have a more difficult time mutating resistance to. And here's a quick diagram of the differences between generations of tetracyclines, moving from the first generation at the top to third generation and fourth generation eravacycline at the bottom. And we have the functional groups here that are responsible for evading these resistance mechanisms. Safety profile of eravacycline, we also have incidence of all adverse reactions between 2 and 8%, so relatively low incidence, including infusion site reactions such as rashes, nausea and vomiting, and diarrhea. All of the warnings associated are class effects with tetracyclines and so carried over as a member of this new class, including anaphylaxis, hepatotoxicity, photosensitivity, and superinfection. Of note, all tetracyclines and this new fluorocycline are demonstrated to cross the placenta and accumulate in teeth and bones, leading to permanent discoloration in infants, and also are metabolized by CYP3A4 in the liver. So be careful with inducers and inhibitors within that class. The landmark trial for eravacycline was entitled IGNITE-1, which is assessing the efficacy and safety of eravacycline versus ertapenem in complicated intra-abdominal infections investigating gram-negative infections treated with eravacycline. So it's a kind of a long-winded trial name. The objective of this trial was to evaluate the efficacy and safety of eravacycline versus ertapenem in complicated intra-abdominal infections requiring surgical or percutaneous intervention. And this trial was designed to show non-inferiority, not superiority. This was a phase 3 randomized double-blind study and analyzed test of cure 25 to 31 days after the first dose with follow-up at 38 to 50 days after the first dose. And it was performed in 541 hospitalized patients, 18 years old or older, with evidence of systemic inflammation and associated pain. And the organizers randomized 1-to-1 eravacycline to ertapenem within those 541 patients and dosed eravacycline 1 milligram per kilogram every 12 versus ertapenem 1 gram every 24 hours for at least 4 days. The results showed a clinical test of cure and an adverse reaction rate both shown to be non-inferior. So eravacycline is non-inferior to ertapenem in cure rate and safety profile for complicated intra-abdominal infections. Later trials included IGNITE 2, 3, and 4. IGNITE 2 and 3 are interesting because they both failed to demonstrate efficacy for complicated urinary tract infection. IGNITE 2 versus levofloxacin and IGNITE 3 versus ertapenem. So we are unable to recommend eravacycline for urinary tract infections at this time as a result of those two trials. However, IGNITE 4 was able to demonstrate non-inferiority of eravacycline versus meropenem for complicated intra-abdominal infection, so it reinforced the results of IGNITE 1. So we have a quick summary, mechanism of action for eravacycline. This is similar to the effects of other tetracyclines. Eravacycline binds the 30S ribosomal subunit and therefore preventing amino acids from being incorporated into bacterial proteins. It is approved for complicated IAIs, dosed 1 milligram per kilogram IV Q12, and is only available as an intravenous solution. 50 milligram vials go for $63.60 out of pocket, and 100 milligrams go for about $117. And it is restricted at UW per infectious disease or attending or fellow pager. And the final antibiotic I would like to talk about is cefidrochol or Fetroja. So in terms of mechanism for cefidrochol, the general mechanism is identical to penicillins, as it is still a beta-lactam antibiotic. There are 5 existing generations of cephalosporin with a widening spectrum. The 5th generation covers MRSA as well as multi-drug resistant strains. Siderophores are molecules with similar chelating properties as iron, and cefidrochol takes advantage of this property in its structure. Siderophores are produced and released by bacteria to facilitate ion influx and create differentials across the membrane through charge and concentration. One unit of a siderophore binds iron and another interacts with the bacterial membrane receptor itself. Researchers have discovered that synthetic beta-lactams with siderophores have low minimum inhibitory concentrations via versus gram-negative bacteria, including Pseudomonas. And that's because these antibiotics are able to abstract and bind iron from the environment, which then binds to the ion transporter and bypasses the defenses of the cell. So the cell takes it in despite the fact that it's actually something that's going to harm the cell by disguising it as an iron ion. And this is known as the Trojan horse strategy, exploiting the ion transport for penetration and avoiding the intrinsic resistance patterns of the bacteria itself. In that way, cefidrochol is known as a first-in-class siderophore cephalosporin, even though it has the same mechanism within the cell. This new research in the early 2000s on siderophore cephalosporins led to evidence of efficacy against Pseudomonas with a good pharmacokinetic and pharmacodynamic profile. Therefore, cefidrochol was approved for gram-negative hospital-acquired pneumonia and ventilator-acquired pneumonia, as well as complicated UTIs. The structure overall is similar to cefepime, with a quaternary ammonium group that is also identical to the one that's bound to cefepime. And it also has an additional aminothiazole ring that lends its broad gram-negative activity. And a carboxylic acid group in carbon-7 leads to permeability in the outer membrane. At UW, this is restricted to an attending or fellow via consult or pager, similarly to eravacycline, and costs $239.09 per 1 gram vial, so also very expensive. Cefidrochol has a very unique spectrum versus gram-negative stains. It targets Enterobacteriaceae, Acinetobacter, Pseudomonas, Stenotrophomonas, as well as Burkholderia and Haemophilus. Of these, the first 4, almost all strains of Enterobacteriaceae, Acinetobacter, Pseudomonas, and Steno are sensitive to less than 4 mg per liter of cefidrochol, so a relatively low MIC seen with those first 4 species. Cefidrochol is a promising agent for CRE pathogens in that 3 consecutive studies from Europe and North America have shown great in vitro and in vivo efficacy. And this is the first approved cephalosporin of note that can treat Acinetobacter. An indication was also added in 2020 for hospital-acquired pneumonia caused by these following species, and there has been no cross-resistance seen between other cephalosporins despite an identical mechanism within the cell. And within complicated UTI patients, 72.6% of infection resolved by 7 days after treatment and 54.6% with alternative antibiotics. So we actually were able to see superiority here, and I'll talk about those trials in a bit. In vitro research have also shown cefidrochol to be 100 times more stable to carbophenamases than ceftazidime is. So this new generation cephalosporin, 5th generation and beyond, now proving that it's more susceptible than 3rd generation agents. Structurally stable as well versus Klebsiella producing carbophenamase, that all of these strategies equally develop resistance to 3rd and 4th generation cephalosporins. So this bypasses those resistance mechanisms as well as evading efflux pumps. So noting that we have a zwitterion like cefepime, very similar structure, and we know that these penetrate better to deal with both gram-positive and gram-negative pathogens. The main distinction that's different in the structure between cefidrochol and earlier cephalosporins comes at position 3, which lends it its siderophore properties. These oxime and dimethyl groups lend to stability to beta-lactamases and overcome the low permeability. Active transport also ensures that this is readily available in the periplasmic space to act on penicillin-binding proteins, since the bacteria are actively taking up this molecule once it binds to iron. All of these are changes from older cephalosporins and contribute to cefidrochol's continued stability versus beta-lactamases. This was also the first beta-lactam to overcome all 3 of the main mechanisms of resistance seen in beta-lactams, including beta-lactamases, efflux pumps, and porins. And of note, not even the previous antibiotic we've discussed, meropenem vaborbactam, is able to bypass all of these mechanisms on its own. Cefidrochol's most common adverse reactions, its incidence was marked as between 3 to 5%, so very, very rare, including infusion site reactions, diarrhea, constipation, and skin rash. Warnings come by class in addition to, so we have hypersensitivity, which is a beta-lactam class effect, neurotoxicity as associated with other cephalosporins, and superinfection, including C. diff-associated diarrhea. So this was a pretty interesting landmark trial that compared cefidrochol versus high-dose extended infusion meropenem for the treatment of gram-negative hospital-acquired pneumonia. And this was a randomized, double-blind, phase 3 non-inferiority trial. The objective, of course, was to assess the efficacy and safety of cefidrochol versus meropenem for hospital-acquired pneumonia. This was performed at 76 centers across 17 countries. All patients were over 18 years old with hospital-acquired pneumonia, and the pneumonia had to be documented gram-negative, so a gram stain had to be performed beforehand. Cefidrochol was given in 3-hour IV infusions alongside meropenem, 2 grams every 8 hours in the control group for 7 to 14 days. And interestingly, all of them received intravenous linezolid for 5 days or more. So a severely drug-resistant organism must have been suspected in order for linezolid to be added. These groups were stratified by infection type and acute physiology and chronic health evaluation score, so Apache 2. 148 patients received cefidrochol and 152 received meropenem. And 86% of them had a qualifying gram-negative pathogen in their cultures. Results of this trial, all-cause mortality at 14 days in the intention to treat population was shown to be non-inferior for cefidrochol versus meropenem. And in adverse reactions were also seen to be very similar between groups. The most common adverse reaction in cefidrochol group was UTI with an incidence of 16%. And in the meropenem group, the most common reaction was hypokalemia. And only 1% of patients in each group discontinued due to antibiotics. So of those, 15% of the patients in each group continued despite the fact that they had an adverse reaction. So the adverse reaction was largely not significant enough to warrant changing the agent. So sofidricol was shown to be non-inferior to high-dose extended infusion meropenem in all-cause mortality at 14 days in gram-negative hospital-acquired pneumonia, and this showed similar tolerability between groups. And my final summary for this agent, for sofidricol, we have a mechanism of action as a first-in-class siderophore cephalosporin with a unique Trojan horse-like penetration mechanism. Currently, sofidricol is approved for hospital-acquired and ventilator-acquired pneumonia, as well as complicated urinary tract infections. It's dosed 2 grams intravenously every 8 hours, formulated as an intravenous solution, and very expensive, costing $239 and change plus 1 gram per 1 gram vial, and it's restricted at UW. And I have our final learning check question here. A 64-year-old male patient acquires Pseudomonas aeruginosa pneumonia during a long inpatient stay. He has a history of hives and wheezing to meropenem, but has tolerated cephalosporins in the past. No past isolates are available and susceptibilities are pending. What would you do if the team asks you for a recommendation? So we have options for cefepime, sofidricol, ceftriaxone, and gentamicin. So clicking through to the correct answer here, we have answer A, cefepime, as the patient will likely tolerate it and it will empirically cover Pseudomonas. As for option B, sofidricol has very broad coverage with a very expensive agent for multi-drug resistant pathogens. This might be unnecessary at this point and may contribute to continued resistance, as we may be leaning towards an agent that's too broad for our purposes. Ceftriaxone also will not cover Pseudomonas, so option C would be off the table. And option D would lead to gentamicin. We would suggest against that use due to potential toxicity and need for therapeutic drug monitoring, especially if an active cephalosporin is available. So finally, key takeaways for innovative antibiotics. Each of these antibiotics was diagnosed to circumvent resistance mechanisms that are becoming more and more prevalent in modern medicine. The most common targets and the most common pathogens that are responsible for those resistance mechanisms include MRSA, gram-negative rods, including carbapenem-resistant Enterobacteriaceae, and vancomycin-resistant Enterococcus. These agents are all tightly restricted here at UW due to broad spectrum and high prices, and are largely non-inferior rather than superior to agents that are older and less expensive. As a result, all of the agents that I've discussed today are only indicated under very specific conditions. And these conditions can include a wide variety, including appropriate pathogen and susceptibilities. It would have to be an appropriate line of therapy. We would have to make sure that the patient is able to afford the medication if transitioning to oral therapy on an outpatient basis, and that the agent is available inpatient and on our hospital formulary. So at UW, it is important to always consult ID if you have any questions about this, pager number 3333, to identify narrower and more cost-effective alternatives if you are considering recommending one of these agents in practice. And I'm more than happy to take any questions. I have my references listed here.